Kia ora everyone. Welcome to this video where I'm going to step through the Tauranga Girls College Level 1 Science Examination for Derived Grades 2020, the Genetics paper, and this video is going to contain question 1. If you're from another school, don't gleefully expect this to enable you to cheat in your own school exams because our examination is written just for our school. But feel free to use it as revision. So, question 1. Question 1 is about the coat colour in cats, and we've got agouti or tabby and non-agouti, and the information in the question tells us that the tabby or agouti is dominant over the non-agouti. So this question, if we look a bit further, there's a lot of context setting as normal. It is a question that's actually more about your ability to understand these terms in bold. And this question often has chromosomes in it as well. So we're just going to add chromosomes in because it's going to help our ability to explain these terms. It does allow you to draw a diagram, and it says that's optional. My advice is if it's space for a diagram, then do a diagram. Diagrams always help you to organise your ideas and to get them across more clearly without waffling. Here is the diagram that I would draw. A pair of chromosomes, each one being a length of DNA. A homologous pair of chromosomes is where each one has been contributed by one of the parents, this one by the biological mother and this one by the biological father. And each of these pairs has the same genes in the same place. So the coat colour gene would be in the same place on each of these homologous chromosomes. So even though the coat colour gene is in the same place, we might have different alleles. That's not spelt great. So this could be the agouti allele, and this could be the non-agouti allele. So this diagram here would be the one that I would draw in my space for in the question given to me. What would I write in here? So I'm going to define these terms. So a gene, to start off, is a section of a chromosome or a piece of DNA, and this piece of DNA has a certain sequence of bases. This is a really important phrase, sequence of bases, and this codes for one protein and therefore one trait. In this case, so I always like to write in my question, in this case, coat colour for cats. So the gene is the coat colour gene. We do have different versions of this gene. Some of my class wanted to say that we have 2 versions of every gene. Now just remember, we simplify everything in level 1, so there could be more than 2 versions, but in this case we have different versions of the gene called alleles, and alleles have a slightly different sequence of bases. Which give a slightly different version of the protein and therefore a different phenotype. And it's good here to explain that a phenotype is the observable characteristics which are coded for by the DNA. DNA. So in this case, the 2 phenotypes, which I could add in here, were agouti and non-agouti. So what have I defined in here? I've definitely got gene, I've definitely got allele. I haven't really defined DNA, so I could just write a definition for DNA. That DNA is a molecule or a macromolecule that provides the genetic information for an organism. And you could go a bit further and you could say that it's composed of sugar phosphate backbone, 4 different bases, and in a double helix shape. That would be really good. So we've got that, we've got that, we've got that, and we've done phenotype. We haven't given the genotype. So I might say here that the genotype is a description of the organism in terms of its alleles. So a description of the alleles. And let's use these alleles in our answer. So AA, big A, little a, little a, little a are the possible genotypes. And I probably would say that these two code for the same phenotype. I kind of want to talk about dominant and recessive here, but I'm not going to because I don't have enough room. And that is... Sorry, just a little interruption there. So I don't have enough room, so I'm not going to talk about dominant and recessive, but I'm going to talk about the fact that that's agouti and that's non-agouti. And that's probably all I need there. So that's question A. Now let's go over here and look at question B, which is continuing about coat colour in cats. First thing is to give the genotypes of 3, 4, 7 and 8 and complete the Punnett square. So this actually, if you can do this and get it right, then this is going to set you up for the rest of this. So let's look at this first. So 3 and 4 are here, and 7 and 8 are here. Thanks, Isis, you'll be in my video now. So let's start with 7 and 8 because they are the easiest ones. Because if we look here, 7 and 8 are non-agouti, and non-agouti, coming back over to here, is coded for by recessive alleles. So we should know that to show the recessive phenotype, we need to have 2 recessive alleles. And then that's a pretty straightforward Punnett square to complete. This one's not quite so easy. 3 and 4 both show the dominant phenotype. So we know they must have at least 1 dominant allele. But the other one could be dominant and it could be recessive. In this case, we can work it out. And we can work this out in one of 2 ways. We can either look at their parent. Parent 1 is little a, little a. And as we get 1 of each allele from each parent, this one had to give this child a little a. We can't work this out for number 3 because number 3, we don't know who the parents are. However, we do know that 3 and 4 had a child with little a, little a. So this is probably the better one, where both 3 and 4 must have a hidden recessive allele to give to offspring 10. So now we know that parent 3 and 4 are both heterozygous. So we can use this to complete this Punnett square. Most of you did this really well. Okay, so there's the start. Now we have to explain why the offspring of 7 and 8 must have the same phenotype as their parents, whereas the offspring of 3 and 4 do not all have the same phenotype. And I've kind of gone through that in my head with my thinking here. So what I need to say for 7 and 8, and I need to label 7 and 8, so there was a few students who still think that I'm a mind reader. You must tell me who you're talking about, otherwise I can't read your mind. So 7 and 8 both show the recessive phenotype. Therefore, both must have 2 recessive alleles, as even 1 dominant allele would result in the dominant phenotype. So what we can see here is that if both parents only have recessive alleles, then all of their offspring must only inherit recessive alleles. Therefore, with only recessive alleles to pass on, all offspring inherit recessive alleles and therefore show the recessive phenotype like their parents. Their parents. Okay, so be really clear here. We want to end up saying that all of the phenotypes of the offspring is the same as the phenotype of the parents. So just wind up with that. So that was actually probably the easier part of the question. Now for parents 3 and 4. We have to explain why their offspring do not have the same phenotype. So this one here is a non-agouti or plain offspring. Okay, and if we look at 3 and 4, they have one offspring that is non-agouti, right? Here's our offspring of 3 and 4 up here. So we have to explain how we know that 3 and 4 are heterozygous. So I'm going to start with that. 3 and 4 are both heterozygous, and I'm just going to use their offspring. We know this as they have an offspring with the recessive phenotype. That's non-agouti. I don't even know if I'm saying this right. Sounds right, I don't know. This means that both parents have a recessive allele to pass to this offspring. So that's number 10. As both parents are agouti, I really don't know how to say that, I should look it up. They must also have at least one dominant allele, i.e. both are heterozygous. That's the important bit there. Now that I've explained how I know they're heterozygous, I can answer the question that they do not all have the same phenotype because the heterozygous parents show these 4 possibilities in their offspring, of which 3 would be agouti and 1 would be non-agouti. So I would just write that down there. So now I'm just explaining what I see in that Punnett square. So I think that's the end of question 1. Yep. All right, I'm going to wind up there and come back with question 2 soon.